The nervous system is built from two major types of cells, neurons and supporting cells. Here is a motor neuron and the supporting cells that insulate its axon. Let's review the various parts of a neuron and their functions. The cell body of the neuron is the part that houses the neuron's nucleus and most of its cytoplasmic organelles. Dendrites convey signals toward the cell body. The axon conducts signals away from the cell body and toward another neuron or toward an effector, such as a muscle cell. The axon is composed of the node of Ranvier, the myelin sheath, and the synaptic terminals. The node of Ranvier is the point where signals can be transmitted along a myelinated axon. Impulses jump between these points. The myelin sheath produced by Schwann cells is the material that insulates axons that convey signals very rapidly. Synaptic terminals relay signals to another neuron or an effector, such as a muscle cell.
What part of a neuron relays signals from one neuron to another or to an effector?
This baseball player springs into action, his nervous system rapidly processing information in a network of nerve cells or neurons. Each neuron receives input from one or more cells. In response, the neuron may generate an electrical signal known as an action potential that travels down the length of the axon. Let's take a closer look at the plasma membrane of an axon. Even without an action potential, the axon is a busy place with many ions moving across its membrane. Much of this ion movement is driven by the sodium-potassium pump. Using energy from ATP, sodium-potassium pumps actively transport sodium ions out of the cell and potassium ions in, creating an uneven distribution of charge across the membrane. Some potassium channels are open all the time, allowing potassium ions to leave the cell. As a result of these ion movements, the inside of the cell is negative relative to the outside. This condition is called the resting potential. The membrane of an axon is also packed with gated ion channels that open and close during an action potential. At resting potential, the gated channels are closed. If a stimulus changes the distribution of charge across the membrane sufficiently, the gated sodium channels open. Movement of sodium ions across the membrane makes the inside of the cell more positive. This reversal of the charge distribution causes the gated sodium channels to close and the gated potassium channels to open. As potassium ions move out of the cell, the original charge difference is re-established across the membrane, closing the gated potassium channels. This sequence of events is called an action potential. The sodium-potassium pump restores the distribution of ions back to their previous levels at resting potential. Let's look at how the action potential is conducted along the axon. As the change in charge difference across the membrane spreads from open sodium channels, other sodium channels farther along the axon begin to open. The original sodium channels close and adjacent potassium channels open. As potassium ions move out of the cell, the original charge difference across the membrane is restored, and then the potassium channels close. Meanwhile, new sodium channels open, followed by the opening of new potassium channels and the closing of sodium channels. In this manner, the action potential is propagated along the axon of the neuron, eventually reaching another cell. The information carried by this action potential will be processed with other information, permitting this baseball player to make a spectacular catch.
Spectacular catch. Oh, I see. A voltage gated sodium channel. Uh, sodium in. No. Sodium out, potassium in. Because it's 2 potassium in, 3 sodium out. Non-gated. If it's non-gated, I would still assume that it's the same. Oh, I see. Suppose that an artificial non-gated potassium channel could be inserted in the plasma membrane of an axon. Does the... What? The membrane potential should impede it.
The membrane of an axon is also packed with gated ion channels that open and close during an action potential. At resting potential, the gated channels are closed. If a stimulus changes the distribution of charge across the membrane sufficiently, the gated sodium channels open. Movement of sodium ions across the membrane makes the inside of the cell more positive. This reversal of the charge distribution causes the gated sodium channels to close and the gated potassium channels to open. As potassium ions move out of the cell, the original charge difference is re-established across the membrane, closing the gated potassium channel.
Let me see what I have. So maybe they're... I'm gonna double check because because in the overshoot right here, when it gets to this plus 30, that is when this thing closes. So at this point in the falling phase, sodium should be closed, potassium should be open. Then here, potassium still needs to be open because it needs to... Okay, maybe this is it. Which of the following characteristics determines when the refractory? Yeah, it's the voltage-gated sodium.
Let's look at how the action potential is conducted along the axon.
Wait, we already
As the change
saw
in
this.
charge difference across the membrane spreads from open sodium channels, other sodium channels farther along the axon begin to open. The original sodium channels close, and adjacent potassium channels open. As potassium ions move out of the cell, the original charge difference across the membrane is restored, and then the potassium channels close. Meanwhile, new sodium channels open, followed by the opening of new potassium channels and the closing of sodium channels. In this manner, the action potential is propagated along the axon of the neuron, eventually reaching another cell. The information carried by this action potential will be processed with other information, permitting this baseball player to make a spectacular catch.
An action potential moves along an axon sequential opening of voltage-gated sodium channels. The diagram below shows voltage-gated sodium channels separated by short distance in the plasma membrane of an axon. The left panel, only A is open within a short time, B also opens. Which statement correctly describes what caused the second one to open? This is kind of true. After the first channel opens, the movement of sodium ions, both inside and outside, alters the ion distribution across the membrane near the second channel, causing it to open. That's really the answer. What? It's gotta be this. But I don't play. Oh. Bro, it's gotta be that one. At location, the membrane potential from a positive okay, so this is the overshoot phase. I guess I would guess D. Wait, the membrane potential changes from positive to negative? Okay, sure, it's the falling. It's stage 4 of the repolarization. Wait, what? Maybe it's F. Because I feel like it's E though. Like it's gotta be E. This is actually confusing, bro. Wait, what? Okay, so F is. Then what the heck could E be? Dude, I don't know how this is so freaking laggy. It's crazy. Okay. So this is F for sure because this is exactly what that is. The sign changes at C. At which point along the axon does the peak in membrane potential occur? D. G is resting. Probably at location A. This is actually probably B. And then this is probably C. The potassium gates close in the overshoot, which is super negative. D is definitely not when the gates are closing. I'm actually so confused. Oh, I think it's this. Okay, that makes sense. For sentence 1, you were given two pieces of information. The membrane potential changes sign from positive to negative, so it's the falling, and the falling phase. B is in the falling phase. Oh, you know what? I think we gotta switch these. Wait, what? In the diagram, A, B, and C represent 3 points along a vertebrate axon where electrodes were implanted to detect action potentials. Under normal conditions, when the neuron produces an action potential, the potential passes through A, then B, then C. Suppose, however, that an action potential is artificially triggered at the point indicated by the red arrow. What sequence would the action potential pass? It's got to be B. Oh my god, dude, this website is so fucked.
Our brains are constantly processing information, which requires communication
All this stuff is public assets.
between billions of nerve cells. Signals are passed from a sending neuron to a receiving neuron at a junction called a synapse. An action potential in the sending neuron travels down the axon until it reaches a synaptic terminal. The narrow gap between the synaptic terminal and the receiving neuron is called the synaptic cleft. The synaptic terminal of a sending neuron contains numerous vesicles filled with neurotransmitters, chemicals that carry information across the synaptic cleft. When an action potential reaches the synaptic terminal, the vesicles fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters affect the receiving neuron, changing the distribution of charge across its membrane. Let's take a closer look. An action potential is propagated down an axon by the opening and closing of sodium and potassium channels. When an action potential arrives at the synaptic terminal, it causes the opening of calcium channels, shown in green. Calcium ions enter the synaptic terminal through the calcium channels. Calcium ions bind to the vesicles containing neurotransmitters. This causes the vesicles to fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters diffuse across the synaptic cleft and bind to receptors in the plasma membrane of the receiving neuron. Here, the receptors are ion channels that open. Ions move across the membrane, changing the distribution of charge across the membrane. The neurotransmitters are quickly removed from the synaptic cleft, ending their effect on the receiving neuron. A single neuron can receive signals from many sending neurons. The signals arriving from the axon on the left are excitatory. They make the receiving neuron more likely to generate an action potential, as indicated by the green glows. Signals from the axon on the right are inhibitory. They make the receiving neuron less likely to generate an action potential, as indicated by the red glows. The two sets of signals cancel each other out, and no action potential is generated. Now, only excitatory signals are transmitted, and the receiving neuron generates an action potential down its axon. The signals that pass between these and countless other neurons constitute our thoughts and coordinate our activities.
Look at that laptop. Look at that laptop. Look at that laptop. Chemical synapses transmit information from the sending to the receiving. The release of neurotransmitter into the synaptic cleft and the resulting changes in membrane potential of the postsynaptic cleft all depend on the presence of several different types of gated ion channels and the distribution of these channels in the pre and postsynaptic cells. Alright. Actually, axon hillock might not have any, right? No, axon hillock definitely has it. Did we get it right? Okay, we got it right. The cell body doesn't have any. Okay.
A single neuron can receive signals from many sending neurons. The signals arriving from the axon on the left are excitatory. They make the receiving neuron more likely to generate an action potential, as indicated by the green glows. Signals from the axon on the right are inhibitory. They make the receiving neuron less likely to generate an action potential, as indicated by the red glows. The two sets of signals cancel each other out, and no action potential is generated. Now,
Wait,
only
didn't we already
excitatory
watch
signals
this?
are transmitted, and the receiving neuron generates an action potential down its axon. The signals that pass between these and countless other neurons constitute our thoughts and coordinate our activities.
The binding of neurotransmitter to ligand-gated ion channels in the postsynaptic membrane causes these channels to open. As soon as the neurotransmitter is removed from the synaptic cleft, the ligand-gated ion channels close. In the brief time these channels are open, ions are able to diffuse across the postsynaptic membrane down their electrochemical gradient. The result is a postsynaptic potential, a brief change in the membrane potential of the dendrites and cell body of the postsynaptic cell. There are two types of postsynaptic potentials: excitatory postsynaptic potentials and inhibitory postsynaptic potentials. In excitatory potentials, excitatory because it makes it more likely that the axon of the postsynaptic cell will trigger an action potential. Conversely, IPSP is inhibitory because it makes it less likely. Okay. Place the phrases into the appropriate bins depending on which type of postsynaptic potential they describe. This actually might be both. I think it's possible that this and this one are over here. Oops. But I don't know. Wait, what? Somehow that got flipped. Oh, that was right. Yeah, that's what I thought. Summation of postsynaptic potentials at the axon hillock. Hillock! Hillock! The figure below shows a single postsynaptic neuron that has synapses with 4 other neurons via the... Wait, what? What? Oh, this is cool. Oh, that's cool. Is that on GitHub or some stupid fucking... Obviously, it's on some stupid fucking Git host. I don't want GitLab. I hate GitLab. Fuck GitLab. I want to star it. It's like, people don't fucking get it. There's actually a point. Now, that's kind of cool, actually. Actually, that's kind of cool. This is pretty sick, though. Why are there so many different graphs? Wait. Let me read this out loud. The graph below shows the membrane potential measured at the axon hillock of the postsynaptic neuron. Each target indicates a change in the membrane potential. Each target. In the membrane potential at the axon hillock caused by a postsynaptic potential at one of the 4 synapses. Summation occurs when 2 or more postsynaptic potentials overlap in time. That is, one postsynaptic potential begins before the membrane has returned to resting potential after a previous postsynaptic potential. When such overlap occurs, the membrane potential measured at the axon hillock is the sum. Place the labels under the graph to indicate which presynaptic neuron produced each change in the axon hillock's membrane potential. Assume the same number of ligand gate opens at each synapse. To review the effect of distance. Oh, really? You gotta look at the distance? Oh, that's crazy talk, man. So then C has to be E1. Then E2 is this. Then this one has to be I1. Do. Which examples correctly illustrate the cooperation between motor and nervous system to maintain homeostasis in a... I feel like all of these. It seems like both of those two are negative feedback, though. But I don't know if this is probably not it either, because that has nothing to do with the brain. Oh, okay, because the hypothalamus. Okay. Okay, so that makes no sense. What? Wait. Oh, shoot. All right, good. We will come back to sensory systems after we finish sensory systems 2.
So in our last lecture, we talked about how in order
I'll be right back.
to generate a neural impulse, we need to accumulate a series of generator potentials, a series of little brief depolarizations accomplished by quickly opening and closing ion gates. And so as these ion gates enter, these brief little depolarization events then, if they add up to threshold, will cause an action potential which will then be propagated and conducted down the axon. We also talked about how to accumulate enough of those little depolarizations, you can hit one collection of gates to open those little ion gates multiple times to accumulate enough to make it to threshold. Or you can have multiple groups of gates opening that are converging on that neuron to more rapidly accumulate those depolarizations. And so this trade-off then between convergence, how many receptors do you have converging onto the same neuron, is related to this trade-off then between acuity and sensitivity. That if you've got a lot of these ion gates that are opening simultaneously, that makes it very sensitive. But you lose acuity because then you don't know where that stimulus occurred. Was it stimulating all those gates or was it just stimulating one of those gates repeatedly? Whereas with fewer gates, then you know that if that neuron is stimulated, it came from gates in that location. And so this trade-off then between acuity and sensitivity with the convergence of receptors, in the case of sensors, of receptors on neurons. Now in the case of sensors, instead of being stimulated by neurotransmitters released in the synapse from excitatory neurons, the gates are being opened in response to some other physical stimulus. And so we're transducing some other sort of signal, other than a neurotransmitter signal, into these little opening gate events, these little depolarization events, that hopefully will add up in the generator potential to threshold. And we talked about the mechanism of how this works in various ways. One way is to have receptors binding to chemicals from the environment instead of a neurotransmitter chemical binding to that receptor, some other substance, but the same mechanism. By binding to that ligand, the receptor is opening ion gates. Or mechanoreceptors, these proteins, these receptor proteins are in the membrane of the cell. So if you distort the membrane of the cell, that causes these receptors to change shape in such a way to open the ion gates to allow ions to flow. And again, these are specific receptors. We're not just creating holes in the membrane. And then in the case of thermoreceptors, these proteins are sensitive to temperature. So you change the temperature, you change the shape of the protein. By changing the shape of the protein, it causes these ion gates to open. So all of these are some of the mechanisms then where a protein in the membrane of these sensory neurons can respond to some other physical signal with opening ion gates and accumulating depolarizations. But now when we talk about responding to light or other forms of electromagnetic reception, sometimes the mechanisms get a little trickier and sometimes we don't really quite understand exactly how they're working. So let's start with light, photoreceptors, and the sense of detecting light that we call vision. And so in the vertebrate eye, our eye, we have photoreceptors called rods and cones in a structure called the retina that are able to transduce the energy of the electromagnetic spectrum that we call light into neural impulses. And so what we call visible light spectrum, that's anthrocentrically referring to our visible light spectrum. As we'll talk about, many other animals have a different visible spectrum. But our visible spectrum then again in this spectrum of electromagnetic radiation, and we talk about the wavelength, so energy traveling is waves. So the short wavelengths down here at 380 nanometers is where we get into the violets. and that goes through the different spectrum up to 750 nanometers, which is in the reds. So things beyond our vision that are shorter than 380 are ultraviolet. Things that are longer in wavelengths than red, than 750, are infrared. So those are the visible spectrum. Then is what our retina responds to. And so obviously the ability to detect light is incredibly important. Although again, we anthropocentrically kind of put importance on it because it's so important to our species. Some other species it may not be as important as some of the other senses. But to be able to detect, where is your food? Where is your other organisms that perhaps want to make food out of you, right? Detecting these things, finding mates. Right? So locating objects, locating your position. So knowing where you are and where you need to go. And then some generalized activities that some animals just in general are more active in light than they are in dark. It's called photokinesis. And so that's why you go in the kitchen, turn on the light, and all the cockroaches start scurrying around because they're in the light. They're more active, and they just randomly move around or chaotically move around until they find a dark place and they stop moving. They don't know where the dark is. They just keep moving until they find dark and then they stop. Or phototaxis, where you actually are moving towards the light or away from the light. So now your movement is directed in direction. So obviously you need to be able to detect light to be able to have these sorts of behaviors. And then it's also really important, sometimes even more so than vision in many organisms, just to know what time of year it is, what time of day it is. And this is what we call photoperiod. And so different times of the year, we have different proportions of day and night. And so by knowing those proportions and recognizing the changes, the days are getting longer, the days are getting shorter, organisms, not just animals, but all organisms can use this sort of thing to therefore alter their behavior to conform to the seasons that are approaching. So these are either daily rhythms, diurnal rhythms of certain activities at different times of the day, or seasonal, what time of year. So that also requires this detection of light and processing that information. So in terms of types of photoreceptors, so the types that we have, that vertebrates have, is called the ciliary type. And so these are modified cilia. So we have a structure that would give us a cilia. This is more like the stereocilia of the hair cells than the motile cilia. So these aren't moving. They're these rigid structures that hold this erect region up here. And then the pigments, right? So if we're responding to light, we need a pigment that's going to absorb that light energy. The pigments are bound in membrane. So if you want to be sensitive to light, you need lots of membrane. And so there's little discs of membrane in this modified cilia to give you lots of pigment exposed to the light. So the light would come shining through this thing, and as it goes through there, it's bound to hit pigment on the way through all those different layers of membrane. So that's a ciliary type photoreceptor. So our rods and cones that we have in our eye are both ciliary type receptors. Interestingly, the cnidarians also have these type of receptors. What's that for an analogy? Whereas most invertebrates have a microvillar type. So again, microvilli, we talked about like the surface of your gut epithelium to increase the surface area. So now it's structurally that type of projection from the side from the cell. And typically it's coming from the side of the cell instead of from the top of the cell. But again, you got all these little fingers of membrane sticking out with pigment in those membranes. And so the light, as it travels through those layers, is bound to hit pigment on the way down there. And so this is the microvillar type of photoreceptors, which you find in most invertebrates other than the cnidarians. Okay? And so this collection of microvilli is called a rhabdomere. So that's a rhabdomere that are microvilli with light pigment, light absorbing pigment in them. So here's looking then again at rods and cones. So from the vertebrate eye, so these are both in ciliary type receptors. And so in the case of the rods, you can see these are taller and thicker, more pigment to make them more sensitive, where the cones tend to be shorter and more tapered, which gives them the ability more to localize the light better. So here's an actual electron micrograph of the retina. So you can see these rods here being fairly tall and flat, where the cones fairly small and pointed. So as we'll talk about, this is going to come back to our ideas of acuity versus sensitivity. Of acuity, you've got being able to detect little points of light, where sensitivity, you've got a bigger area receiving light. There's more to it than that, but we'll come back to that. Now, interestingly, in the vertebrate retina, partly as a byproduct of how our nervous system develops, remember, as we just talked about in lab, we go through neurulation, and our nervous system develops from that hollow dorsal nerve tube. And so our nervous system then is developing as branches off of that hollow tube. And so when it enters into the structure, the cup structure of our eye, these neurons, these series of neurons enter in and then point away from the light. So it's somewhat surprising that the photoreceptors are actually pointing away from the light. And so when the light comes through, it's got to go through. So here's the actual neurons that are coming from the optic nerves. These are called ganglion cells. And then these synapse onto these bipolar cells. And the bipolar cells synapse onto the actual photoreceptors, the rods and cones. And so the light has to go through these other layers of cells before they reach the actual photoreceptors. Although we'll see there's actually some exceptions to this. So some people have questioned, why would, it seems like an odd design, why would you have your photoreceptors pointed away? And so when people are not so arrogant and thinking, oh, that's stupid, they start realizing, of course, form and function, that there's a very good reason to have the photoreceptors in close association with this epithelial layer at the back of the eye. Okay, so it's these layers of neurons here, that's the actual retina. The back of the eye here is this epithelial layer in the back layer. And so there's been people who actually study the eye then realize this is absolutely vital and important way that the eye is built that makes the eye much more efficient. And so for one reason, so there's a whole series of different reasons why this association of photoreceptors with the epithelial layer at the back of the eye is so beneficial. That for one is that these cells at the back of the eye are pigmented themselves to absorb light, not for photoreception, but to prevent light scattering, because the light's bouncing around inside your eye. Then when if you stimulate a photoreceptor, you don't know where it came from. And so by having this dark pigment, then any light that doesn't hit a photoreceptor is absorbed, and it's only going to be light that's coming in through your pupil that's going to be stimulating the rods, not light that's bouncing around inside the eye. So this light absorption part of the epithelium is important. Then there are also to nourish these cells, because they're really active and they're doing a whole lot of work here. That nourishment in terms of sugars for energy source, vitamins that you need to create the pigments, those are being supplied to the photoreceptor cells from these epithelial cells, as well as gas exchange. There's also, when these pigments get worn out, they can digest away the tips, and so you're making new membranes, these plaques, new membranes of pigment at one end and removing the old ones at the other end. So this constant removal of old worn out pigment plaques also is accomplished by this epithelial layer. And then the paracrine communication that we talked about. So there are these growth factors that are regulating the health of these cells, that are regulating when they need to grow, when they need to be phagocytized. So all of this is happening with this layer of epithelia at the back of the eye, helping the retina be healthier and much more functional. So it's not at all a bad design. Okay, so we said that this membrane-bound pigment then is where we're actually absorbing the light. How are we transducing, though, that absorbing light into a neural impulse? And essentially what happens is there is a pigment molecule, again, a derivative of vitamin A, called retinine. And so it actually, when it absorbs light, changes shape. So in the dark, it has a sort of bent form here, which is called a cis isomer. When it absorbs light, it transforms itself into this more linear structure, which is called a trans isomer. So it changes shape in the presence of light. There are proteins that this pigment is bound to called opsins like optical. And so these opsins then, it's like you're binding to a chemical receptor, only you're already bound to it, but just this ligand is now changing shape. And so when the ligand changes shape, the protein changes shape. When the protein changes shape, it can regulate ion gates. So how ingenious is that? And so here's, again, a opsin. So in your rods, the pigment is rhodopsin, so it's a particular one. So here's the retinol pigment, and then here's the opsin protein embedded in the membrane of these discs. And of course, lots of membrane, there's lots of pigment here. So light coming through here is likely to hit that pigment. And so it changes, again, the ionic permeability, briefly opening and closing these ion gates by these light hitting these things and turning them into trans isomer. And then there are enzymes in the cell that convert it in the dark back into the cis isomer. So that's why when you go from a dark to a light environment, at first it looks really bright until it adapts, because the light's rapidly turning this. When you go from a light environment to a dark environment, it takes longer for your eyes to adapt, because this reaction in the light is rapid. The enzyme reaction is slower. It takes longer for your eyes to reset to the dark. Okay, so this is called the bleaching reaction. So just like you leave a pigmented cloth out in the sunlight and it bleaches and turns lighter, in the same way we have the pigment changing in response to the presence of light. But we have enzymes that can reverse that change, unlike your clothes that you left out and got faded in the sun. Okay, so in the rods, it's rhodopsin. In the cones, it's still going to be the retinine, but it's going to be different proteins, so it's going to be other opsins. Now, what's interesting, so far in our transduction mechanisms, we've talked about different ways to open the sodium gates and depolarize the cell to get to threshold. Interestingly enough, in most cases, not all cases, but in most cases, in the photoreceptors, it's the opposite. It's actually inhibiting them. It's creating inhibitory postsynaptic or inhibitory signals. Well, that seems odd. Why would you respond to a signal by inhibiting it? And partly it's a way to become actually more sensitive. So stay with me here. So essentially what it is that the ion gates in these photoreceptors are normally open. So they're constantly leaking ions, and so they're constantly firing themselves. They're usually stimulating themselves. Okay, we talked about this in like the hair cells, where there's a background rate of alpha effect.
So the plates are here. They're here, whatever.
Now, what's interesting, so far in our transduction method, in the photoreceptors, it's the opposite. It's actually inhibiting them. It's creating inhibitory postsynaptic or inhibitory signals. Well, that seems odd. Why would you respond to a signal by inhibiting it? And partly it's a way to become actually more sensitive. So stay with me here. So essentially what it is that the ion gates in these photoreceptors are normally open. So they're constantly leaking ions, and so they're constantly firing themselves. They're usually stimulating themselves. Okay, we talked about this in like the hair cells, where there's a background rate of how fast they fire, and you can either make that rate faster or slower. So there's a background rate in how fast these are firing. Now, which means they're secreting a neurotransmitter all the time. But the neurotransmitter they're producing is an inhibitory neurotransmitter. So they're inhibiting the bipolar cells. Right? Remember, we went from ganglion cells, bipolar cells, to photoreceptors. They're inhibiting the bipolar cells from firing. And so when you turn off the photoreceptor, you stop inhibiting the bipolar cell. And therefore the bipolar cell fires and stimulates the ganglion cell. So when you turn off the photoreceptor, you stop inhibiting the bipolar cell, and therefore the bipolar cell fires and stimulates the ganglion cell. Okay, so it's actually a way to make it more sensitive to light, where you have this background firing rate and you're turning that off. So again, in the dark, then the photoreceptors are releasing an inhibitory neurotransmitter that hyperpolarizes the bipolar cells. So they're not firing. So the light inhibits the photoreceptors, therefore you're inhibiting the inhibitor. Right? So double negatives make positive. So that releases the bipolar cells to leak their own ions, produce an excitatory postsynaptic potential in the ganglion cells, and transmit action potentials down the optic nerve to the brain. So this flow of ions in the dark, where in the photoreceptors normally their gates are open, is called the dark current. So the dark current means the rods and cones are all kind of chronically leaking sodium ions and therefore constantly stimulating themselves to fire. But when they fire, they're releasing an inhibitory neurotransmitter. So the light blocks the dark current, which blocks the inhibitor. Okay. So here's trying to illustrate that here. So in the dark, again, these neurotransmitters are, so the pigment is in the cis conformer, and therefore this thing is constantly depolarizing and releasing inhibitory neurotransmitters, inhibiting the bipolar cell.
Inhibitory. Glutamate inhibitory.
Conformer, and therefore this thing is constantly depolarizing and releasing inhibitory neurotransmitters, inhibiting the bipolar cell. In the light, the pigment changes shape, the gates then close, so it stops releasing the inhibitory neurotransmitter. So now the bipolar cell releases its neurotransmitter. Okay, so the light is blocking the inhibitor, therefore allowing the bipolar cell to fire. And this is, again, makes it so sensitive that in many cases a single photon of light can cause a response. Okay. So that's what we mean by the dark current. So we're inhibiting the inhibitor in the presence of light. Okay, so that was our transduction mechanism of how do we actually get a nerve impulse in response to light. And so light is actually blocking a nerve impulse of an inhibitor, therefore allowing another cell to release its neurotransmitter. Now in terms of structures, again, we talk about different things that organisms are using light for. And so in many cases, the structure of their eye will relate to what do they need the light for. Is it for forming an image, or is it for detecting the presence of light in terms of timing and photoperiodism? Is it the direction of light for navigation? Those sorts of different functions of vision will determine the structure of the eye. And so, for example, if we look at the flatworm, when we looked at the flatworm in lab, we talked about its eyes that it had in its head. And in these cases, we have the eye, the shape of the eye is this cup-shaped structure with pigment in it called screening pigment. And then here's your photoreceptors in that cup. And so by being cup-shaped, it gives you, well, having photoreceptors, you can now detect the presence or absence of light. So now your photoperiodism can be detected in that way alone. But the cup gives you this directionality. So that if light's coming from this direction here, it's going to be striking these photoreceptors, but this one over here is going to be shaded out. Whereas if light's coming from the other direction, then this photoreceptor is in the shade, and these are being more stimulated. So having a cup-shaped eye allows this directionality to know where is that light coming from. Or conversely, where is it not coming from. And so this pigment then prevents the light from coming from the other direction, just when these flatworms are really flat, and stimulating them from the other direction. So they've got to come be exposed to the light that's not being shielded by the screening pigment. Okay, so these eyes that are not used for image formation, but they're using for detecting the presence and direction of light, we call an ocelli. So ocelli are these, some people call them simple eyes, that aren't necessarily for image formation, but their primary function is high sensitivity to the presence of light and some ability to detect the direction that the light is coming from. And so we can see in different animals this trade-off then between the sensitivity, because the flatter it is, the more light's going to be shining on your photoreceptors, the more exposed your photoreceptors are going to be to light. The deeper the cup, the more shading it's going to be. So you're, again, sacrificing sensitivity because you're not allowing as much light in, but gives you better ability to detect the direction of that light. And so we can see this, that if you're in an area with lots of light, then you can get away with a deep cup. If you're in a low light environment, or you don't really need to know the direction so much, then you can have a shallow cup. So this is sort of the direction here. Here you'd have no cup, which no animal has. So this is a model of flat. No animal has a flat eye. But here, like we saw in the flatworm, and here in this limpet as well. A limpet is a snail that's got a really flat shell. It's got a fairly shallow eye. Whereas here you go into some other snails, it's got a deeper cup, so now you get more directionality, more ability to tell the direction of light, but less sensitivity. It's harder for the light to make it in there where the photoreceptors is. And then here we go down into, we actually have a range of different snails that have this whole spectrum of different eyes here, depending on where they live, as well as there's some annelids that have this kind of eye. Also, here we also get into some gastropods as well as snails that start having these deeper cups. So the nautilus here, the nautilus again is pretty deep where it's fairly dark, so you don't want the hole to get too small. Okay, so here we've got a cup. Here the cup is actually even impinging over to start giving you what we call a pinhole eye. Now, one way to be able to give you even better directionality without sacrificing that opening is to add a lens. So a lens helps you focus light. So now even with a bigger opening, you can still focus that light on a point. So now having a lens allows you to be able to pinpoint the direction of that light without sacrificing the size of the opening. Okay, so we get this whole gradation then between how deep is the cup and the presence of the lens, where you no longer have to have a really tiny hole to give you good directionality. Okay, so these pinhole eyes, adding lenses, and the ability to focus the lens is another mechanism that allows for improved, not just detection of light and direction of light, but now you start getting into image formation. And so with that pinhole eye, you can now, the reason you can start using image formation is because you have different cells receiving different parts of that image. So here, if you're looking at this tree, I guess it is here, that the light coming off the top of the tree is hitting photoreceptors here. Off the middle of the tree is hitting photoreceptors in the middle. Off the bottom of the tree is hitting photoreceptors here. So different photoreceptors are responding to different light coming from different parts of this object. And therefore the brain can take all that information processed together into an image. And so the better you can focus where that light came from, the better you are at creating an image in your brain of the source of that light. By source of light, right, doesn't mean it's glowing, it's reflecting light. And so you're able to see it because the light reflecting off of this object is impinging on photoreceptors at very specific places inside the eye. Okay, so the smaller that opening, or the better the lens is for focusing that light, the better you are at forming an image. So we said that if you have, again back here, that if you're really, for example, looking at these are marine snails, the deeper you are, the less light there is. You can't afford to have a small opening if you're down deep because there's just not enough light. You need to have that more open cup. But what's nice is to be able to adjust the size of the opening. And so we can do that with a structure called an iris. So the opening in the eye is called the pupil. If you have an iris, that means that it can actually change the size of the opening. Okay, so it's a series of smooth muscle, in the case of vertebrates, that can constrict and retract. So you have some muscles that open it up, some that pinch it together, so you get both peripheral and circular muscles here, allowing you to change the size of the iris, therefore change the size of the pupil. So now under highlight conditions, you can make that opening smaller, and therefore get more precise location of where that light is. Whereas if you're under darker conditions, you can dilate that pupil and let more light in under low light conditions. And you're sacrificing the ability to form a precise image, but at least you can detect the light. So here you can balance your needs for acuity versus sensitivity. Okay, so once we add a pinhole eye with a lens and with an iris, we now call this a camera eye. We call it camera eye because that's how we build cameras. Cameras have these features, right? We're imitating a camera eye when we build a mechanical camera. And so here, for example, is comparing how we build a camera compared to how our body builds an eye. So again, the opening for the light to enter in the camera is called the aperture. In your eye, it's called pupil. Right? So if you need to let in under low light conditions, you want to take a picture under low light conditions, you need to increase your aperture, or your f-stop on your camera. And so you have a diaphragm control that controls that. Whereas in the eye, we've got iris muscles that control the size of the pupil. Now to focus the light even better, we use lenses. And so we use glass lenses. In a good camera would have glass lenses. That's a series of these lenses that are used to focus the image on the film or on the photosensitive chip. And we have a series of things that are focusing the light in the eye. We have a cornea, that curved cornea, and then these liquids of different density here, the aqueous and vitreous humor, and then a lens here that are again allowing us to focus light on the retina in the back of the eye. So the actual transduction mechanism in an old film camera would be the photosensitive chemicals on the film, and an electronic camera would be the little chips on your photosensitive chips on your chip, in your camera. And then in your eye would be the photoreceptors, the rods and cones in the retina. And then, as we talked about, we don't want light bouncing around. If we want to have good location of where that light came from, we want a dark surface. So the inside of the camera is typically painted black, and the back of your eye is pigmented with that dark pigment in the epithelial cells, so the light's not bouncing around. So you know that if light hits that spot on your retina, it came from that direction, and it didn't come from just bouncing around inside the eye. Same thing inside the camera. Okay, so these are parallels then in how we build a camera to how your body builds an eye. One of the main differences is how do you focus it? That if an object changes distance, then you need to be able to adjust how that light is focused on the retina to keep it focused. And so in the camera, you've got to have multiple lenses, and you change the distance between those lenses because they're rigid lenses. Your body makes a better lens. It makes a flexible lens. You can actually change shape. So you have to actually change the shape of the lens to focus it.
That's actually crazy. That's pretty crazy.
So here's some different examples of eyes we find in different animals.
I'll be right back.
Has some interesting variations. So here we have, again, light bends, like anything that travels in waves bends when it goes from one density media to another. Right? So we talked about the acoustic lens of the dolphin where it can focus sound waves. In the same way with light waves, that when it transfers from one area where the media is a certain density to another density, that causes the light to bend. Now for an animal that lives in the ocean, a marine animal, the difference in density between the inside of its tissues and the seawater isn't so different, so it needs a bigger lens to focus that light. Whereas the difference between air and body tissues is much greater, so you can get away with a flatter lens to accomplish that same focus. So we typically see a bigger, rounder lens in a fish than we would in a terrestrial vertebrate. As opposed to, again, just a pinhole camera where the only thing that's focusing it is the size of the hole. But again, you can't focus it quite as well. Here we're focusing onto a single photoreceptor. Here, that same amount of light, actually even less light, so you're getting a lot more light here focused on that, which helps the sensitivity. Here, that much light is hitting more photoreceptors. So having a lens greatly improves the ability to focus that light. Now, if you look at arthropods, remember, are dicozoans. They don't have that holodorsal nerve tube. So their nervous system, right? Remember, neurons develop from ectoderm, but their ectoderm doesn't go through forming that neurulation, that holodorsal nerve tube. So it's forming from that outer layer of their body. So you get here, this ectoderm develops into this neural tissue, and then other layers of ectoderm develop into the other layers. And remember, their whole body is covered with that chitinous exoskeleton. And so their cuticle, that outer part of the cuticle, is actually forming the cornea of their eye. Okay, so we get this cuticular cornea, and then they form a little crystal in here, crystalline lens. But they tend to be more fixed in shape. They can't really focus them. Okay, so they're again trying to do it by, instead of going through different liquid media here, it's pretty much a solid media that's bending the light. Now, if you've ever played with optics, you know that if you want to bend light, you can use lenses or you can use mirrors. And interestingly enough, in some of the bivalve mollusks, things like scallops, they actually use mirrors instead of lenses to bend the light, which is pretty darn cool. So here, for example, in the scallop eye, this bivalve mollusk, that the light comes through, again, they have that surface eye, right? These are not chordates. So the pigments are laid down similar that they were here in the arthropod. But as it goes through that layer of pigment, it then bounces off a mirror in the back of the eye, and that curved mirror focuses the light on the individual photoreceptors. So they use mirrors to focus the light instead of using lenses to focus the light. It's an interesting little difference in accomplishing it. So speaking of the arthropod eye, so remember, for non-vertebrates and non-cnidarians, we have those microvillar type photoreceptors. And arthropods in particular, and some annelids have this also, and some mollusks have this, they have what we call a compound eye. So a compound eye is like a whole set of eyes all packed together. And instead of being a deep cup, it's more a bowl shape that bulges outward. And so each of these little facets then is its own little cornea and lens. Right, so here's the cornea, as we just talked about previously, developed. It's from the actual cuticle that would form the exoskeleton, and the rest of the body is from the cornea over the eye. And then under that, here's that crystalline cone that collectively forms the lens of each of these little segments, which is called an ommatidium. So ommatidium is one of these little units, visual units. Then from the lens structure here, we go down to this tube, so this tubular eye, that's lined with these long microvillar photoreceptors. And there's 8 of them. So there's 8 of them in a ring shape here, or forming a cylindrical shape, and all those little microvilli are pointing towards the center of that ring. And so that's called the rhabdom, the light path. So those little microvilli, the rhabdomeres, from these 8 cells are overlapping. So now the light is focused down this channel to hit all these photoreceptors. Okay? So that's in each of these ommatidium. Okay, so the rhabdom is the light path, and 8 of these photoreceptors in one ommatidium. And then you've got hundreds to thousands of these ommatidium forming one big compound eye. Okay, so now light hits here, so it's like we talked about in the cup-shaped eye, the chambered eye, we call that. In these, again, part of the image is going to hit this ommatidium, part of the image is going to hit this ommatidium. So they are image-forming eyes, but a different mechanism, where each photoreceptor's got its own, each set of photoreceptors has its own lens, as opposed to one lens focusing on particular photoreceptors. So a different way of accomplishing the same thing, the same process. And so it's unlike in the movies where they show, the insect's view of something, you're seeing multiple views of the same thing. It's not. It's like your eye. Different ommatidia are looking at different parts of the image, and the brain is assembling those different parts into an image. So they are really good at forming images. Not quite as good as the chamber eye, but they're excellent at detecting motion, at changes. Because if light's coming from bouncing off of something here, and then all of a sudden it moves and it's sitting over here, it's a really distinct change in terms of which ommatidia being stimulated. And so the arthropod eye is really good at detecting motion. That if something moves, if something changes in its visual field, it's really fast at responding to that. So it's faster at responding to changes in the visual field than the chambered eye. Whereas the chambered eye is a little bit better at forming images, the quality of the images, the precision of the images, the acuity of the images.
That's pretty cool.
So that's the compound eye, the microvillar type photoreceptors arranged into ommatidium. So again, you see these in arthropods, both terrestrial and aquatic, as well as in some annelids and some bivalve mollusks. So again, anatomically deriving from different structures. So how do you get the exact same structure in unrelated organisms? It happens to show up more than once. It's convergences from the and stuff. So again, here just looking back grammatically at some of these different eyes we've talked about. So here in the nautilus, again, nautilus lives in fairly deep water, and so it's got this no lens and a fairly open eye because it's living in the dark. You need to have a big opening there to be able to respond to whatever light there is. Whereas you get into an octopus that lives in shallower water, it's got a shallower cup and it's got a lens to be able to focus light much more precisely. Okay, and the same sort of thing we would then see in a terrestrial vertebrate, although again, a terrestrial animal can be a much shallower lens than the big lens you need in an aquatic animal. And then here's the compound eyes, like we saw in the previous picture. So here's in a feather duster worm, so an annelid worm, having those things, as we talked about when we looked at feather duster worms in that little video clip, where they're very sensitive. If something comes at them, they can really quickly pull themselves back in. If they're not really forming images, they just need to know something's coming. Whereas here, again, an aquatic arthropod, there's less light, so a little less ability to separate little parts of the image. So usually you're a little less ability to form images. So here in the case of, actually down here, in the case of krill or the lobster, a lot more light needed to stimulate, or you need to accumulate a lot more light to be able to stimulate individual photoreceptors, where in this terrestrial arthropod, this dragonfly, which have lots of ommatidia, it takes a smaller visual field to be able to stimulate the photoreceptors because there's just more light in the terrestrial environment. And then here's the scallop. So here's a little blue-eyed scallop here. Those are the eyes on the edge of the mantle. And so that's using the mirror to focus the light. That's the mirrored chamber. Okay, so again, chambered versus compound eyes, and then lens focusing versus mirrored focusing. Now, we mentioned the cnidarians having these ciliary type photoreceptors like vertebrates do. And it's actually, to think of the jellyfish as having eyes at all is surprising to many people. Or you might think it's got just basic, yes, presence, absence of light, which some jellyfish, that's what they have, the ocelli. But then you look at something like the box jelly. The box jellies are the class of cnidarians that include things like sea wasps and the ones that have really potent stinging cells. And they're called box jellies because they typically have 4 corners to the edge of their bell. And at those 4 corners are these sensory structures called ropalia. So at each corner there's a ropalium. So ropalium singular, ropalia plural. And each ropalia has 6 eyes. So that means it gives you a total of 24 eyes on these things. And those 6 eyes are different. So here's one Ropalia. So here's the Ropalia, here's a blow up of the Ropalia. It's got a pair of pit eyes. It's got a pair of slit eyes. And the advantage of a slit, like a cat's eye, is that it gives you more variety, meaning to dilate and constrict it. So if it's slit, it's easier to open up into a bigger opening and then close it off to a smaller opening. So it gives you a bigger range of pupil size for different light conditions. And then it's got a pair of camera eyes. So with the lens, with the pupil, with the iris, one pointing upward and one pointing downward. So pointing upward and downward for, again, with a nerve net type nervous system, it's like, how is it processing all this information? This elaborate eye it really has not been studied very well other than to be amazed that they exist in terms of how is this animal processing visual information and how complicated is the visual information that it can process. Okay, so this is again just sort of summarizing the vertebrate eye structure, where again we've got our cornea, which is the main thing that's bending the light, focusing it on the retina, and then we go into these different chambers here of different liquids, and then the lens here, which is doing the final focus. Sort of fine-tuning focusing. And the retina on the back of the eye, the optic nerve comes off over here. So the part directly behind the center of the cornean lens is a region called the fovea centralis, and that's where you have the highest light impinging. And where the optic nerve comes in, there are no photoreceptors there because that's where you're plugging the system in. So that's your blind spot. Okay, and then again we have this camera eye where we can adjust the size of the pupil under different light conditions. And again, it's the top of an image that we're looking at is actually on the bottom of our eye. The bottom of the image is the top of the eye, and your brain then sorts it out the right way. In fact, they've even done these experiments where if you give somebody a pair of glasses that flips an object upside down, first it looks upside down, but after a while your brain's able to sort it out and start to make it right side up again. Now if you take the glasses off, everything looks upside down. Your brain has to adjust back to where it's supposed to be.
That's crazy.
And the image itself is formed by the brain. The brain is using this information to form an image. So again, we said with the advantage of a lens, the type of lens we have as opposed to that cuticular lens of invertebrates, is that we can change its shape. And so this ability to change the shape of the lens to keep an object in focus, because as an object comes from far away to close in, right, it's changing, the light is changing from it, right? When it's far away, right, my hand looks huge now, so that light is coming to a very small part of my eye, whereas it comes closer now to my face, it's looking smaller because it's now changing the direction of where the light's coming from. So the ability to keep that image in focus as it comes close to you or away from you is called accommodation. And accommodation is achieved by this rapid mechanisms of neural images being processed by the eye and these muscles called ciliary muscles that stretch the lens out. So the lens is naturally round. These muscles flatten it out. And so when an object is far away, you want it flat so the light's coming straight in. As an object comes closer to you, that lens needs to round out. And so those muscles relax, allowing the lens to become rounder and rounder and keeping the object in focus on your retina the whole time. And so we don't even notice it happening. It's such a fast reflex where the brain can determine whether an object is in focus or not and whether or not it needs to change the shape of the lens. I mean, just absolutely amazing ability to do that. Now, of course, the brain is assuming that your eye is the right shape, that under the correct shape, then it's adjusting the shape of the lens so that its light coming in through your cornea is going to be focused on the retina in the back of your eye. Okay, and so for an object that's greater than 20 feet away, that's why we say 20/20, the object light should be coming in straight in from that, and so your lens should be at its flattest and therefore be focused on the retina. But as you grow, if your head grows and funny and your eye becomes oblong, elongated, now it's focusing the light where your retina should be, which means it's focusing in front of the retina instead of on the retina. So by the time that light reaches the retina, it's out of focus again. So to correct for that, we give you concave lenses that help spread the light out so that now it will be brought to focus on the retina. On the other hand, if your eye got too short, now the light never had a chance to focus before it hit the retina. So now you need to have a convex lens to start focusing that light before it even hits your lens, so it will focus in the back of your eye. Okay, so your brain is working that you're going to have a normal shaped eye. If your eye's not the right shape, it's not going to be focusing correctly. Now, the astigmatism typically means there's been some aberration in the shape of your cornea or the shape of your lens, and so now different light coming from different places is being focused differently. So then you need a lens that's got different thicknesses in different places. It needs to be a little more complex structure. That's an astigmatism. Okay, so far we've looked at the ability to work with the the quantity of light, right? So sensitivity to low light versus being able to focus light more under high light conditions. But another aspect of perceiving our visual world is the quality of light. That spectrum of light that we call the visible light spectrum, there were a whole variety of different wavelengths there, from 380 up to 750 nanometers. And so if we can see the quality of light and tell the different wavelengths of light, that gives us the ability to distinguish even more detail in the environment. So color vision is an aspect of acuity, that if we have two dots close together, it's a lot easier to tell them apart if they're different colors, right? If you can tell that they're different colors. If you can't tell they're different colors, then you're only on the space between them. It's the only thing that can help you with acuity. Remember, acuity is the ability to see details. And so not all animals have the same color mechanisms that we do. And so, for example, I look at a tiger and those bright orange stripes it's got, and going, how would a predator ever catch any prey with those bright stripes? Well, the answer is that most mammals are colorblind. And so to a deer, a prey item, that tiger, those stripes are blending in with the shades of background. So they're not showing up distinctively to typical animals the tiger would be hunting. So let's get into this idea then of color vision and how that relates to acuity. And interestingly, this is a phenomenon of mammals more so than most animals. Most animals have pretty good color vision. As we'll talk about, some of them have color vision that blows ours out of the water. But most mammals are nocturnal, so most mammals are going for sensitivity instead of acuity. And color vision is part of acuity. If you need sensitivity, then you sacrifice acuity. You sacrifice color vision for sensitivity. So since most mammals are nocturnal, then they need to be able to have high sensitivity. They sacrifice the acuity, the color vision, for the sensitivity of night vision. Now, there are exceptions among the mammals. The old world primates, so the primates of Africa and Europe, Europe, Africa, Asia, do have color vision. They tend to be more diurnal, active during the day. And so since they can see color, they also use color. And so many of them have pretty good color vision. Animals the tiger would be hunting. So let's get into this idea of that bright stripes. Well, some acuity vision is an aspect of acuity, that if we have two dots close together, it's a lot easier to tell them apart if they're different colors, right? If you can tell that they're different colors. If you can't tell they're different colors, then you're only on the space between them. It's the only thing that can help you with acuity. Remember, the acuity is the ability to see details. And so not all animals have the same color mechanisms that we do. And so, for example, I look at a tiger and those bright orange stripes it's got, and going, how would a predator ever catch any prey with those bright stripes? Well, the answer is that most mammals are colorblind. And so to a deer, a prey item, that tiger, those stripes are blending in with the shades of background. So they're not showing up distinctively to typical animals the tiger would be hunting. So let's get into this idea then of color vision and how that relates to acuity. And interestingly, this is a phenomenon of mammals more so than most animals. Most animals have pretty good color vision. As we'll talk about, some of them have color vision that blows ours out of the water. But most mammals are nocturnal, so most mammals are going for sensitivity instead of acuity. And color vision is part of acuity. If you need sensitivity, then you sacrifice acuity. You sacrifice color vision for sensitivity. So since most mammals are nocturnal, then they need to be able to have high sensitivity. They sacrifice the acuity, the color vision, for the sensitivity of night vision. Now, there are exceptions among the mammals. The old world primates, so the primates of Africa and Europe, Europe, Africa, Asia, do have color vision. They tend to be more diurnal, active during the day. And so since they can see color, they also use color. And so many of them will actually change colors depending on their social position in the troop, in their sexual state of, are they receptive or not. So the color of the face, the color of the scrotum, the color of the females changes to announce both their position in the troop and their susceptibility or receptibility to breeding. So the cone system then is our acuity system. So that's helping us see detail. And one of the ways that it does that is being able to see the difference in different colors. So cones have much less convergence than do rods. Remember, convergence gives you sensitivity. Rods are all about sensitivity, so they have high convergence. Cones are about acuity, they have less convergence. So having less convergence, they're less sensitive. So cones give you better acuity, rods give you better sensitivity by their convergence. Now, adding to that low convergence color vision allows you to see even more detail, because we said that we can tell it's easier to tell things that are close together if they're different colors, or if we can perceive that they're different colors, than if we can't. If they look to us like the same color, it's harder to tell them apart. It's harder to see details. So under high light conditions, those rods get bleached out, and you're left with those low sensitivity cones. And so that bleaching reaction bleaches out your rods because they're just too sensitive. So now they're not, right? They're all not producing their. Yeah, they're not working. They're not responding. So only your less sensitive cones are going to be active under high light conditions. Now, in our eyes, we have 3 different types of cones. And so this gives us what's called the trichromatic theory of color vision. Tri, chrome, color. So we have our 3 different types of cones that respond to different wavelengths of light differently. And so the brain can process this in terms of when light hits that cone, in terms of different cones respond differently to different lights, the brain takes that information, puts it together to create the color. So all the colors of the spectrum that we see are variations in how these 3 types of cones respond to that color. So another way of saying it is that these are the 3 primary colors of transmitted light, and all other colors are made by a combination of these colors. It's like in the old days when you had a projection TV, you'd have 3 different projectors. You'd have one that projected a green image, one that projected a red image, and one that projected a blue image. And then all the colors of the image that showed up depended on how those overlapped each other. So other colors are detected as a blend of these 3 colors. So if we look at the spectrum here, so we have this one type of cone that maximally is stimulated by this wavelength of light, but still is stimulated by wavelengths of light in this range. Our second one that's stimulated here, which we usually refer to as green, but it's still overlapped here. And then here, more towards the red end of the spectrum, we have this one here that's maximally here. And so when we start getting out here to the edges of red, this is the only one that's being stimulated. Out here to the edges of blue, this is the only one that's stimulated. And so depending on the wavelengths of light that's hitting you, it's stimulating different cones differently. So out here in this range, you're only stimulating these cones. Here, you're stimulating both these cones a lot and these cones a little bit. Here, you're stimulating all 3 of them, but to different degrees. Here, you're just stimulating greens and reds, then here, just reds. And so your brain is taking all that information about which cones are being stimulated and how strong are they being stimulated, processing that together and giving you the perception of different colors. Brain is amazing. What can we say? Now, we mentioned that fovea centralis is that place directly behind the center of the cornea. So this illustration is kind of odd because it looks like this is the center. This is the optic nerve. That's your blind spot. So the optic disc is where you don't have to see any color. The fovea is where you get direct impinging from looking. When you look straight at something, the light's hitting here. And if you look at close here, you see actually that those ganglion cells and bipolar cells are pushed over to the side. So the light is not going through all those different layers. It's hitting directly on the photoreceptors. And the photoreceptors in the fovea are all cones with no convergence. No convergence, meaning you have one cone under one bipolar cell under one ganglion cell. So no convergence, therefore the maximum acuity. So low sensitivity, maximum acuity. Right? So if you stimulate that cone, you know exactly where that bit of light came from. Okay, so the degree of convergence of the cones is actually 1 to 1 to 1. So one cone to one bipolar cell to one ganglion cell. So no convergence, you have the highest acuity. So when you want to look at the precision detail, you look straight at it. So that the light's hitting right there in the center of your eye, in that fovea. As you go towards the periphery of your eye, then you start getting a mix of more rods and cones, and you start getting higher convergence of both rods and cones until finally you get to the periphery of your eye, and it's all rods, and they have a lot of convergence. So the highest acuity is more towards the center of your eye, the highest sensitivity is towards the edges of your eye. Now, we said that most mammals are colorblind. That doesn't mean that they don't see color. It's just they don't see as many varieties of colors as we do. So, for example, they would only have dichromatic. They only have 2 types of cones instead of 3 types. So here, if we compare our visual spectrum, where we can see all this continuum of colors from red to violet, the dog's is seeing less of that. It's only got the red and green type cones. And so it's seeing, in this range here, where there's just the red cone being stimulated, there's no difference. Where here, where there's overlap between the red and the green, you can see variations because you're seeing differences in how much the red versus the green. And then out here, there is no blue cone, so it's not seeing any color out here where we would start seeing the violets in the spectrum because there's no cones out there that respond to that wavelength. So they see some variation in color here. They see color, but no variation in color here. But they can certainly see, right? They can tell orange from blue, but they can't tell like red from orange. Okay, so that's dichromatic color. So similar in humans that have what we call red-green color blindness. So there are humans that have this sort of vision also, this color vision also, if they have a defect on one of the genes on their X chromosome that prevents them from creating the blue type cones, blue type opsin that would respond to that. Remember, all your visual pigments, they're all retinine on a protein. The difference between the 3 cones and the rods is the protein. Now, again, the reason for this is they're going for sensitivity. So not only do they have less sensitivity in terms of being able to tell color apart, we can't tell that from that, but the overall acuity is less as well. So a dog can see detail at less than 12 cycles per degree, where we can see over 30 cycles per degree. What does that mean? That means in terms of, if you look at your visual field, 1 degree of vision, if you break that up into 30 bits, you can still see that it's individual points and not a solid line. Where a dog can only break up into 12 bits to see a solid line. So where you can see this is all being a series of whites and blacks, to a dog that would look like just one solid smear. You couldn't tell that it was broken up into details. You'd have to be broken up that much for the dog to be able to see that it's actually differences. Right? So that's acuity. So they have lower acuity, we have higher acuity. Right? Because they're going for sensitivity, not acuity. So again, equivalent to 20/80 myopia and nearsightedness. So if you're a nearsighted colorblind person, you see like a dog does. Of course, your brain is still able to process the information that a dog can. So here's again what we mean. So again, if you couldn't tell any color, it would look like this. With a typical mammal dichromatic color vision, you'd see this much variation, right? So you can see more detail, being able to see color. And then with trichromatic vision, you can see even more detail, right? And that really stands out against that background. Okay. Plus, again, the human brain is an incredibly powerful computer. And so we see saturation of color and intensity of color, the sort of velvet complexity of color, much more than any other animal can. So because our brain is processing this information much more powerfully, we can see color much more intensely than other animals can. So only humans can appreciate art, really, because we can only really see those subtle variations. Our brain can perceive that from that visual information. So other animals may have actually better eyes, but we got a better brain. Now, there are animals that have trichromatic or even higher variability in terms of photoreceptors, but they can maybe see a different spectrum than we can. So for example, here's our spectrum again, going from violet to red. The bee, that spectrum is shifted towards the violet. So a bee can't really see red very well, right? So like when we talked about our flower lectures, that red flowers typically are attracting birds more than bees. But they can see down here into what we would consider ultraviolet. So where it's too violet for us to see, the bee can see it. Okay, so they're picking up visual information in the spectrum where we can't. We're picking up visual information in the spectrum where they can't. And so if we look at this flower, this butterfly, we see this much detail. The bee, in addition to this detail, can also see these details, right? Because this is the parts of the flower and the butterfly that are now reflecting the ultraviolet light, right? So they can see these details, plus they may not see the red dots, but they can also see this. So they're seeing differences. So when sometimes we look at a flower, we look at a bug and 2 different bugs, and they look the same to us, they may look different to the bugs. So sometimes we do DNA testing, find out that things that we thought were the same were different. The bugs, they look different. They look different to each other than they do to us. And so this, again, is not a table you need to memorize. This is just examples of variations in visual abilities of different animals. Not that we've necessarily tested their vision, we've just looked at what pigments they have in their eyes, and from that inferred what colors they can probably see. And so again, with a lot of your terrestrial arthropods, they're seeing typically down here in the ultraviolet. Whereas in your aquatic arthropods, ultraviolet doesn't penetrate underwater. So there's no sense being sensitive to ultraviolet. There is no ultraviolet. There's also not very much red. Red doesn't go very far underwater either. So most of your marine animals only see in the blue spectrum, because only blue light's going to penetrate to any significant degree. Where we get down here into, again, most mammals have that bichromatic color vision, except for those old world primates, and also diurnal squirrels. So here's showing blues and yellows in squirrels, but some of your squirrels do have more of a trichromatic color vision. Whereas your New World monkeys, your South American monkeys, are more like typical mammals in having the dichromatic color vision. Now birds, on the other hand, birds have 5 to 7 different types of photoreceptors. They're seeing color spectrums that we have no idea how to comprehend. So birds are seeing color in the world in ways that we can't even imagine. So probably necessary for their ability to find bugs and flowers and fruits and things like that. But who knows why? And probably why they have so many colors themselves. That color is very important, how they communicate with each other, because they can see color so well. And then sometimes there's just things where we kind of scratch our heads and going, what the heck is going on? So the mantis shrimp, for example, the mantis shrimp lives underwater, so there shouldn't be that much variety of color, but it lives in shallow tropical water. Tropical water is clear water. And they have up to 16 different types of photoreceptors. So their visual spectrum is going clear down from the ultraviolet to the infrared, and lots of variations in between. Right? So here's our color vision, our 3 types of photoreceptors and our visual spectrum. Here's their visual spectrum. What are they doing with that? Behaviorally, we don't really see that they necessarily have that great color discrimination, but is it because we're not testing them right? We have no idea. Plus, they can actually see whether light's polarized or not. So like having polarized sunglasses on, makes things look differently. Their eyes automatically do that. We can see depth perception because our brain compares the vision in our 2 eyes, their stereo vision. They can do that with each eye. Each eye gets 2 versions of the image. They can actually do stereo vision in 1 eye. So each of their eyes is doing. They have
How do they do that?
Stocks, and so they can get a 3-dimensional image of an object from different directions. And just, what are they doing with this visual information? No idea. It's just bizarre creatures anyway. So again, this idea of stereoscopic vision and how our brain processes information is that our brain does compare our 2 eyes, right? This is why we have 2 eyes, is so our brain can compare those 2 eyes. And so the version of the eye that your left eye sees compared to the version of the image that your right eye sees. The brain uses that to create this 3-dimensionality. And to make those comparisons easier, it's interesting that it's not like the left side of your brain sees the right side of the right eye and the right side of your brain sees the left eye, but it's the right side of your visual field on both eyes is processed by the left side of your brain. The left side of your visual field from both eyes is processed by the right side of your brain. So it's able to compare those 2 versions. So you see that in the optic nerve here. So here the photoreceptors on the left side of both eyes is going to the left side of the brain. Right? So these photoreceptors go onto an optic nerve that crosses over to the other side, called the optic chiasma. These don't. So these ones on the right side of the brain stay on the right side, where from the left eye, they cross over to the right side. Okay, so the left side of the brain is getting the left side of both eyes. The right side of the brain is getting the right side of both eyes. Okay, so it's going from, and then your visual cortex is in the back of your brain, where it's processing all this information. So by being able to compare those images, you can put together fine differences in those 2 versions of the same image to create this 3-dimensionality, this overlapping of visual fields. And so we typically see then the amount of overlap is related to how important is 3-dimensional imaging to an animal, particularly now we're talking about vertebrates. But in predators who are hunting things and therefore need to know how close the thing is before you jump on it, typically have a flat face with both their eyes facing forward, so there's maximum overlap of the 2 eyes, of the visual fields of the 2 eyes. So the brain has the information to very effectively create a 3-dimensional field. Right? So predatory species, as well as brachiating species that are doing a lot of branch-to-branch type movement, right? We need to know exactly where that other branch is that you're about to jump to. Those animals typically have a flat face with eyes facing forward, much greater degree of overlap of the 2 visual fields. Whereas prey species, it's more important to them to know who's sneaking up behind them. So they typically have a narrow face, their eyes set on the side of their head, so they can see behind themselves. So this is an exaggeration here. It's not that there's no overlap in their visual fields, but their visual fields are allowing them to see both in front of them and behind them at the same time, because they're on the side of this flatter head. Right, so flat in that dimension as opposed to that dimension. But then, vertebrates, our body plan says we only get 2 eyes to work with. You start looking at invertebrates, where instead of being extensions off that hollow dorsal nerve tube, your eyes are developing from your ectoderm anywhere you want them, and you get eyes that are just all over the place. And some of them are just absolutely amazing. So, for example, in jumping spiders, they don't hang out in a web, they move around and hunt, right? And they actually jump on prey. They have 4 pairs of eyes, and they're all doing different jumps. It's like this came out of a hunting design factory. It's amazing stuff. So they have 4 pairs of eyes. So the first part, the anterior medial eyes, so anterior in the front of the body, medial in the middle, have high precision vision. So this is their high acuity vision. So these little kind of seeing very good detail, looking straight ahead of them. Then their anterior lateral eyes, so again more to the side of the front of their head, have these wide overlapping fields of view to give them stereo vision. Because again, they're jumping on prey, they need to know exactly how far where that prey is. Then their peripheral eyes, they've got medial eyes that give them 180 degrees vision on either side of their body, so they can see all the way behind themselves on the sides, and then the lateral eyes, and then medial eyes are looking at in a dome above them, so they're seeing 180 degrees in an arc above them. So they're seeing information everywhere. You can't sneak up on these guys, right? They can see everywhere with high precision vision and stereo vision as well, having all these eyes specialized for different functions. It's a proportionally really big fang, so it's a good thing these guys are tiny. Now, some other specializations even within the vertebrates we can see. So as we mentioned, the fovea centralis is where we have the highest acuity vision, right? We're looking straight at something. Raptors, birds of prey, have 2 fovea. One where you're looking straight at something, and one at the top of their eye when they're looking down at something. So an hawk or an eagle can see in front of them and below them at the same time. So they can see if they cross over prey. And there's also a lens that magnifies the vision coming down below. So it's like 8x binoculars, eightfold magnification binoculars looking down while they're looking straight ahead at the same time. I mean, just obviously their brains are processing all this information. So again, they can fly straight. Ahead, they can detect things below them and pounce on that prey. Okay, so that's if you're specializing for high acuity. Now, if you needed to do that mammalian thing, or that nocturnal mammalian thing of high sensitivity, then let's exaggerate in that direction. And so you've probably noticed that, your cat, a lot of mammals, have these highly reflective eyes. And that's because the back of their eye, instead of having that dark pigmented epithelium that prevents the light from scattering, they actually have mirrored backs of their eyes. That's called the tapetum lucidum, the glowing tapestry. So it's a reflective layer behind the retina. So the epithelial layer, instead of having those dark melanin pigments absorbing light, have these guanine crystals that are reflecting light. So mirrored eyes. So again, present in most nocturnal mammals. Actually, even in other vertebrates besides mammals. So for example, sharks. And so what this mirror does is give you a second chance. So if that light shines on your eyes and it doesn't happen to hit one of your photoreceptors on the way in, it'll bounce off the back and maybe it'll hit one of your photoreceptors on the way out. So it gives you a second chance for your photoreceptors to respond to that light signal.
Crazy.
Missed on the way in, maybe it'll hit on the way out. So this increases the sensitivity of the eye over 6-fold. So again, if you're active at night, sensitivity is more important than acuity. Now we said that dark pigment was because you want to eliminate that bouncing around because it eliminates acuity. So there is a sacrifice here of acuity. Because light is bouncing around the eye, was that light coming in from the outside, or is it bouncing around, in which case you're not quite as good at locating where that light originally came from. So it's tapetum lucidum, this reflective layer at the back of the eye, giving you better night vision but poorer acuity, along with the dichromatic vision, also giving you better sensitivity, lower acuity. And so we'll be dissecting pigs, or virtually dissecting pigs, in lab. Of course, in the preserved ones, you can't really see it too well, but if you did a fresh pig, pigs have this tapetum like most mammals do. So again, there's the cornea of the eye, you remove the cornea, and then there's the lens of the eye under that, you remove the lens. And now, so here's the back of the eye, so here's the retina, which is highly vascularized. And then you peel the retina off and you see this kind of greenish, yellowish green material back here. So those guanine crystals reflect light in the sort of yellow-green spectrum. So this, again, increases sensitivity over 6-fold. So here's that butterfly we looked at before. So in the dark, where we're using our rods because our cones aren't sensitive enough, we would see maybe that much of the image, where the cat, with its dichromatic vision and its tapetum lucidum, would be able to see that much detail. So they can see in the dark much better than we can because of this tapetum. And so another aspect of night vision is having that slit pupil, as we talked about in the box jellies, that allows that opening to vary much greater, where you can close it off during the daytime, because again, they have the high sensitive eye, you need to be able to close it off in daytime, so there's too much light. But at nighttime, you want it wide open. So you can see here how small the pupil is, the small slit during the day, during ambient light. At night, so it's taken with a flash, so it was wide open before you hit it with a flash. Big difference in how wide they can open their pupil. And then the color of the eye is coming from pigments actually in those iris, the muscles of the iris that are creating the dilation of the pupil. So this is a cat that doesn't have a tapetum in one of its eyes, so it has a mutation or some defect where it's not able to put tapetum in one of its eyes when it was developing. So the color of your eyes is typically the amount of melanin in the eyes. So with a little bit of melanin, you're scattering the light instead of reflecting the light. And so just like the sky scatters light, and that's why the sky looks blue, that's where you get blue eyes. Whereas if you have more pigment, then you start getting into browner eyes, because that's the spectrum of light that melanin absorbs. But here, this blue-eyed cat, you have no tapetum in this eye. You have a tapetum in this eye, so you have that blue color from the iris and then that yellow-green tapetum behind it. So that's why one eye was kind of yellow-green and the other one was blue, because this is the blue eye with and without a tapetum behind it. And then when you flash, now you don't have the iris there because the pupils are dilated, so the iris isn't in the way. The flash is going to be reflecting off that tapetum, giving you that yellow-green light, whereas in the eye without a tapetum, it's just the blood vessels in the eye that give you that red eye effect. And again, since we don't have a tapetum, that's why you can flash picture people and you get that red eye effect, because we have blood vessels in our eye, but we don't have a tapetum. Now, we mentioned last time the difference between passive senses and active senses. So passive senses, you're responding to stimuli from the environment, so in this case, light from the environment. With active senses, you're producing the signal yourself. So in this case, some animals do produce light. And so there's again, interesting convergence of the actual biochemistry of this reaction where you get this whole spectrum across different phyla and even kingdoms that are able to produce this light called bioluminescent living light. And so in some fish, they have the enzymes where they can bioluminesce. Others actually will culture bioluminescent bacteria. And so it's actually bacteria that are luminescing, and the fish just gives it a place to live. And so it cultures the bacteria to produce light. And so, for example, these flashlight fish that usually live in deeper water or in caves, they have this little pocket of bioluminescent bacteria under their eye, and then a flap like an eyelid that can cover it over. So they can cover it over when they don't want light, then they open it up and they can shine it. So they can shine this light in front of them. So they're making the light that they're seeing because they're living in the dark, so there's no light around them. Now, just on that subject of bioluminescent, again, it's this huge spectrum of organisms. And we're going to talk about, for example, bioluminescent fungi, of organisms that can produce light. So there's some insects, like fireflies, glowworms, things like that. There are photosynthetic protists, dinoflagellates. So recently, in fact, you go out on a dark night and look at the waves breaking, you see the waves glowing because there's a dinoflagellate called Noctiluca that seasonally gets abundant. And so when you agitate it, they glow. And so when the waves are breaking, you see this glowing. It's pretty cool. When you go diving, sometimes you can actually like draw underwater, wave your hand underwater, you see little glowing trails to excite these dinoflagellates. And then a lot of deep sea fish, mollusks, crustaceans will produce light to lure prey. We mentioned that with our feeding lecture, creating these little glowing lures that attract prey, or to signal mates. We talked about, again, in our feeding lecture, how in some fireflies, which remember are actually beetles, they're predatory species that will flash one pattern to attract
all
mates,
vision,
and then flash the mating pattern of the other species to attract prey.
All vision.
And then some of them use them for what we call counter shading, where they will have a series of these photophores, right, these light bearers, along their ventral surface, and they glow to match the intensity of the sunlight above them. So that way, anything that looks up at them, their belly blends into the sky, because they're glowing at the same intensity. Whereas an animal looking down on them, they have a dark back, and so they blend in with the dark, deep ocean. So it's a way to hide in plain sight. So there's, in the open ocean, there's nothing to hide behind. So you blend in with the light. Now, looking at some photoreception weirdos, or some unusual variations of vision from what we usually think of in our own anthrocentric perspective, that the cephalopod mollusks, the squid and octopus, again, remember they're not chordates, so they don't have a hollow dorsal nerve tube, so their eye develops from their ectoderm and not that neural ectoderm, not from that neural tube. So they're developing very differently from different tissues, but they end up with a structure that looks incredibly like the vertebrate eye, only deriving from completely different structures. And so one perspective, though, that because the pigment cells are deriving from the body wall itself, the photoreceptors are facing outward. So unlike ours, because the optic nerve comes in and then points around, the photoreceptors are pointing away from the light, theirs are pointing towards the light. So that's one of the aspects of deriving it directly from your body wall. As your photoreceptors go outward, you have this cup shape, there's your lens, and then they sort of pack in there. Remember, these are microvillar type photoreceptors as opposed to ciliary. So different type of sensors, different neural development, and yet we come up with something so similar. So they tend to be packed in the back of the eye, looking this sort of pattern like this. But then despite that, they come up with an eye pattern that is incredibly vertebrate-like, or conversely, the vertebrates come out with one that's incredibly squid-like. And so again, here's ours. So here from our hollow dorsal nerve tube, this extension comes up, and then pockets inward from the retina of our eye. So again, since this formed from a pocketing inward of the body wall, the neurons are pointing the other direction. And then the ectoderm derives into the cornea and lens of the eye. In the arthropod, because it's deriving from the body wall, the photoreceptors are pointing outward. So here's those ommatidia, here's that crystalline lens, here's the cornea, the cuticular cornea of each ommatidium of the arthropod eye. Now the cephalopod is starting out with this kind of development, right, the body wall type photoreceptors, but then achieving this sort of body shape, this sort of eye shape. And to do that, it pockets in, then bulges out, then pockets in again to give you a structure. I mean, and again, the photoreceptors are pointing outward. They then do develop a cornea and lens. And so despite this completely different origins embryologically in tissues that are creating the structure, they come up with an eye that looks almost identical to a vertebrate eye. I mean, it's like, a industrial espionage, right, where somebody stole the plans from somebody else and reverse engineered it to their bow plan. The components of the eye are almost identical in completely unrelated organisms. Now, some other oddities that we see in the visual world. So we said that cones are good for acuity, and color vision is part of acuity. But if you're active at night, if you're nocturnal, then you want sensitivity. So how do you achieve color vision and sensitivity?
Oh, that's cool. That's neat. I like that one.
In this case, the color is not necessarily for acuity, but it's for something else. So for example, in the fruit bats. So fruit bats, unlike the insectivorous bats, the one that echolocate, these are feeding on flowers and fruits. And so you need to find flowers and fruits by color, right? So fruits are ripe when they turn color. That advertises their ripeness. Or flowers advertise for their pollinators by color. So how can we increase the sensitivity of our eye and still have color vision? Well, you increase the surface area of your retina. And how do we typically increase the surface area of any sort of organ when we want to increase surface area? We wrinkle it. And so they actually have a wrinkled retina to increase the surface area. Now that's going to inhibit the ability of you to focus for high acuity vision, but we don't need high acuity vision. We're looking for sensitivity here. So we have color vision and sensitivity by increasing the surface area of the retina by wrinkling it. So nocturnal color vision. Then, of course, if you want to not sacrifice your acuity that much, but you really need the sensitivity, you don't need the color vision, you need sensitivity, though. Like in these tarsiers. Tarsiers, whole animal fits like in the palm of your hand, tiny little eyes. But these things are jumping from branch to branch in pitch darkness. They need really good night vision, right? Because you don't want to miss that branch or be able to catch a bug in the dark. Biggest eyes in proportion to their body size of any vertebrate. And so their eyes are so big, though, that they can't move their eyes. So it's not like, I can look over there by just moving my eyes. They can't do that. Their eyes are just too big to move, so they have to move their whole head in order to see. To compensate for that, their neck can actually rotate 180 degrees, so they can turn their head and look straight behind them. Now here's an interesting anomaly. We want a fish that's, again, living out in that open ocean under that sort of mid-ocean range where there's some light, but it's mostly dark. And they want to be able to see prey or predators above them and below them at the same time. So they have eyes that point up and eyes that point down. But wait a minute, the vertebrate brow plan says you only get two eyes. So how can you have a pair of eyes looking up and a pair of eyes looking down when you only have two eyes? And so here we're actually seeing, it's called the spookfish. It looks pretty spooky. Here looking down on the dorsal view of it, we can see these two eyes looking upward. And then here's the ventral view. You can see these two eyes to the side looking downward. Right, so here's actually the back of these eyes looking downward. Okay, so these are called the primary eyes looking upward and the secondary eyes looking downward. How did you get four eyes if you're a vertebrate and are programmed for only two eyes? And the answer is you split your eyes in half. So here's the primary eye is the main eye. So the primary eye is oriented to look upward. So here's the lens focusing light on the retina in the back of the eye. But embryologically, they split their eye. So the part of the retina is over here, part of the retina is over here to the side. But now the lens focusing light over here, what good is this retina here? They take the trick from the scallops, from the bivalve mollusks, and use mirrors. So they have a mirror here that reflects the light coming up from below onto this part of the retina. So the lens, so this is their basic eye is here. So lens focusing light on the retina, but this part of the retina is getting light reflected off this mirror. I mean, how amazing is that? So here's an actual image of the light coming into this secondary eye and that curved mirror focusing that light onto specific photoreceptors on that portion of the retina. How cool is that? Now, as we said, with the non-vertebrates, if you're not a vertebrate, you're not limited to these two extensions off of your holodorsal nerve tube to make eyes. You can make eyes in all kinds of places. And so we call these extraocular photoreceptors. So extraocular, so outside the eye. So photoreceptors not located in a structure that we usually call an eye, i.e. not on your head. And so, for example, insects have really complicated genitals, really complicated reproductive structures. And so one way, because again, with a million species of insects, you want to make sure you're mating with your species, right? You'll be wasting your gametes where they're not going to do you any good. But the genitalia are the back end of your body, and your eyes are on your head. So again, tactily, often these insects will have little kind of feelers on their butts that kind of locate everything. But some insects have figured out a way to do is have what do we call those rear view cameras?
Backup camera.
Like on a truck or a van or whatever, you're backing up, you see rear view. They got cameras on their genitals, so they can actually see as they back into each other and wind up their genitals, so that here's the penis of the male, here's the vagina of the female. That's when you get into the right place. And so here on the male, here's an eye over here. We don't call it an eye, we call it a photoreceptors here, photoreceptors here. Here on the female, and photoreceptors here. Instead of a pair of eyes, actually four eyes, so two on each side, so that they can line up and back into each other in such a way to effectively copulate. So, I don't know if that's someplace you want to have eyes. Another example of extraocular photoreceptors are in some of these deep sea shrimp. So these shrimp are living down in the bottom of the ocean where it's completely dark. There is no ambient sunlight. And so, not surprisingly, they don't have eyes. Okay, you live in the dark, why do you have eyes? But then when people collect them, they notice that there's kind of a little shiny patch on their back. You can see it very well here. This kind of little shiny patch on their back. And when they collected some of these and looked at them, they said those are photoreceptors. So again, typical shrimp eye is that compound eye. Instead, that photoreceptors are spread out in these plates on their back. Okay, so why would they have these if they're living in the dark? So why have photoreceptors and why have them on your back instead of in your head? And of course, at this point, it's all storytelling. It's going on when people bounce ideas around. And that, obviously you have a bigger surface area exposed here by putting your back instead of limiting with your eyes. You're not worried about image formation, but maybe more direction the light's coming from. Although how directional are they if they're a flat plate? So originally people thought that maybe because these things are living on those thermal vents where the hot hydrogen sulfide is percolating through the Earth's crust and creating this community of bacteria that feed on those things, and so that's what the shrimp are feeding on. So maybe that they're sensitive to infrared light created by hot bodies, and that's how they find where these thermal vents are. But then they tested the pigment, saying, "Oh, it's typical shrimp pigment that's sensitive to blue-green light, not to infrared." And so then they're saying, well, maybe it's because some of the bacteria that are growing in these thermal vents are bioluminescent. Maybe that's what they're attracted to. And so that's kind of the latest story. But again, it's a little hard to observe things down there in the bottom of the ocean. So just an interesting observation that they don't have eyes, but they do have photoreceptors spread out on these plates on their back, these dorsal organs. And then we can see eyes in various places. So when we looked at sea stars, for example, we saw that they had eyes on the ends of their arms. Well, it turns out that in sea urchins, they actually have photoreceptors in the ends of their tube feet. So it's like every one of their little tube feet is like this little mobile little photoreceptor. How good of image formation do they have, or directionality do they have? Who knows? Why would you need them on the ends of your tube feet if you're not doing some sort of directional or image formation with them, right? Otherwise, you could just have them in the base of your body. But here you can see they've used expression of these opsins, these pigments, these photoactive pigments, and see the ring around the edge of the tube foot here is where those genes are being expressed. And then there's a nerve that comes down the side of the tube foot, and here's those holes in the ossicles. Remember, we looked at those where the tube foot penetrates through the ossicle, and there's a little notch on the side where the nerve goes through. So it's got some nerve innervating this light-sensitive structure around the edges of its tube feet. So how cool is that? I mean, at this point, we've just noted their existence. We have no idea how they actually function in terms of the biology of the organism. And then this one was interesting. Again, the story, it's all interesting observations, and who knows what's going on. I mean, there's so much to still study out there. But in brittle stars, so another echinoderm, so another one of our classes of echinoderms, the ophiuroids, it was noted that they have photoreceptors in their central disk. Okay, presence, absence of light, right? Well, then somebody else noted that if you look in the electron microscope at the structure of the central disk, the ossicles in the central disk, and it's a series of these little, very smooth, very orderly arranged disks. And then in cross-section, when you look through them, you see that they're located right above where the photoreceptors are. It's like every one of these is like a lens of an eye. And so is the back of a brittle star a compound eye? One giant compound eye. Are they doing image formation? What part of their is their central nervous system sophisticated enough to create images? No idea. And the photoreceptors even have an iris-like chromatophores to shield them during the daytime so they don't get overexposed. Amazing stuff out there. Okay, so that's just some little taste of the varieties of vision, of animals responding to what we call light. Now, there are other spectrums of electromagnetic energy out there besides what we typically call light. And not too surprisingly then, considering how amazing different organisms, unrelated organisms are at responding to light, we would have organisms that can also respond to this other spectra of electromagnetic energy. So for example, warm bodies typically emit infrared radiation. So it's not just heat, but it's a spectrum of radiation that warm bodies tend to radiate. So can you detect infrared radiation? Well, there are some animals that can. So for example, in a couple of different orders of snakes, the pit vipers, which include things like rattlesnakes, and the boas and the pythons, have these pit organs in their face. So the pit vipers have a pair of them on either side. The boas and the pythons have a series of them running the sides of their face. And these, like a pinhole camera, are a chamber with a small opening. So it seems like they're kind of focusing a signal. And the back of that chamber has some sensory cells that respond to infrared radiation. So they're able to tell. Now, how well can they form an image with this? We don't know. Or can they at all? Or is it just sort of general directionality that sort of something warm is over there as opposed to over there? Or can they actually do some sort of image formation with this? Don't really know. Now, why would they want this? Well, remember, these are cochilotherms, so their body is relatively cold. A lot of times they're hunting warm body prey, right? Birds and mammals. And so if you've got a warm body prey against a cold background, it's going to show up. And so maybe that's a way to locate warm body prey. Some other people have speculated that it may be a part of behavioral thermoregulation, that if they're cold, they need to find a warm place to go so their body doesn't chill, since they're not making their own body heat. So it's how they find, look, that rock is warm, that rock is cold, a way to find where to go to behaviorally thermoregulate. So you go camping and you wake up with a rattlesnake in your sleeping bag. It's like, hey, you are a warm place. Now, it turns out that there's one mammal that also has infrared receptors, and that's the vampire bat. We talked about him a little bit. We talked about osmoregulation. So again, the only known mammal that has infrared receptors. And so it's also got these pit organs, these leaf pits around the nostrils. So here's its two nostrils, nares, and then here's these three pit organs that have infrared receptors on. So what are they doing? Well, again, these are looking for blood meals. And they don't suck blood, but they do use these little incisors here to nick a blood vessel and then lick up the blood of their prey. And so you don't want to just randomly be finding some cow and biting it in various places. You want to bite it where there's a blood vessel. So this essentially is how they map out where the blood vessels are on their prey item. So they go find some cow or whatever mammal that they typically feed on is, and they can map out where the blood vessels are and say, okay, bite here. So that's infrared sensing. What about other spectrum of electromagnetic system, like electrical fields? That there are some animals that have sensors, and in that case, we're not really sure how they work, we just know that they do, that can detect fluctuations in electrical fields. And so they can use this to detect prey, for example. So for example, when you're creating all those neurochemical impulses from nerve activity or muscle activity, those create little tiny electrical fields. And so if you can detect electrical fields, you can detect where somebody may be hiding. Or navigation, that if you live in seawater, a current of seawater is ions in motion. Ions in motion create electric fields. So maybe you can use it to navigate. Or there are some animals that create electric fields. So now it's an active sense where you can create an electric field and then feel how that environment is distorted by the environment around you and use that to navigate. Or if you can produce electric fields and detect electric fields, now you have a communication system that you can speak and listen and nobody else can, because unless you can detect electric fields, you can't. Nobody's going to hear you. So again, it seems odd to us because we don't do it, but there are animals that do. Particularly aquatic animals, because air doesn't conduct electricity very well. And so we have an example in mammals, that the monotremes, the mammals that lay eggs, so the platypus and the echidnas, have electroreceptors in their bill, in their rostrum. And so that's one of the reasons why the duck-billed platypus has that duck bill, is that's like a minesweeper looking for electric fields. So when they're hunting, again, these are hunting in water, and usually murky ponds and streams, and at night, so they're not visually hunting. In fact, they close their eyes, they close their ears, and they close their nostrils when they're underwater. So it's like you're in one of those isolation tanks, right? Just close off all your senses and be in tune. Well, they're tuned in with the electrical sense. So all their other senses are kind of closed off, and they're just focused on this electrical sensing. And so they're hunting things like these little crayfish over here, and they're doing such a good job that they're eating half their body weight every night. Using this electrical sense. So they're very good at finding prey in these murky conditions in the middle of the night using this electrical sense. Sharks and rays also have a system where they can detect electrical fields. These are called the ampullae of Lorenzini. So if we look here at the shark, sharks do have nares that they use for smelling, not for breathing, so they have a very good olfactory system. We also talked about their lateral line system that has a branch around the eyes, around the mouth. They can tell vibrations in the environment. And then all over their snout and face, they have these little pits. And those are the ampullae of Lorenzini. So here's a little close-up that goes this little sort of jello-filled canal down to these sensors that somehow are transducing electrical fields into nerve impulses. And so they can use this to hunt prey, to navigate using saltwater and ocean, to hunt prey. Hammerheads, again, have that big flat head. We talked about how it spreads out the nares for olfaction, but also spreading out these ampullae of Lorenzini to this minesweeper. And so these will often swim right over the surface of the sand and detect if there's a fish buried in the sand. The muscles that are just ventilating their gills generate enough electrical field that they can detect that. And they're looking at things down in the nanovolt sensitivity. Right, so we talked about our voltage changes being millivolts. We're going from millivolts to microvolts to nanovolts. They're detecting voltages down to those levels. Also may be important that it's on their snout because, again, when they're biting something, they can't see it. It's the lateral line and the ampullae of Lorenzini that are telling them where to bite. Right, so your olfactory system gets you in the general location. Your visual system gets you closer, but it's your lateral line and your ampullae of Lorenzini that are doing the final zeroing in.
Have an electrical field sensor.
That's why it's speculated that might be why in some of the old movies when people were trying to film white sharks, and the shark would come up and they have a big piece of bait, and the shark would come in, instead of biting the bait, it would bite the cage. But, well, these sharks are so aggressive, they're trying to eat the person in the cage. Well, probably it's zooming in on the bait, but then the electrical field from that metal cage just distorted it, and that's what it did. Now, another animal that's known to use electrical fields for hunting is the lungfish. Lungfish, so-called because they can actually breathe air for short periods of time. And can use that because they live in places where their ponds dry up and they have to get from one pond to another, so they can crawl out of the water and go from one pond to another. But it's been discovered that they do have electrical senses as well, similar to ampullae. And so these people are trying to study, well, how important is that as opposed to its sense of smell for hunting as opposed to its electrical sense? Which is more important? And so what they did is they had some lungfish and some tanks, and they had in the tank these little traps, these little chambers in the bottom of the tank covered with sand. And then they would either put prey in the little traps to see if the lungfish could find them. And okay, yeah, they can find them. Okay, are they smelling the prey, or are they looking using electrical sense? And so here you would see if you're above the line then where the live prey is, so you're using live prey. So here you put live prey in there, and yes, they would find them in the right place. So they would have two chambers, in other words. And they put the prey in one and not the other, and say, would you pick the right one or the wrong one? And so the bar above the line is showing that it's picking them in the right chamber. Then they said, what if we put a live prey in one chamber and then had a pump that was taking the scent and putting it to the next chamber? And said, no, they're still looking for in the right place. So even though the scent was in the wrong chamber, they're still finding the prey in the right chamber. Okay, what about if we electrically shield that chamber so that it can't emit an electric field? Then all of a sudden it's going to the wrong place. Then it can't detect the live prey and it's going where the smell is. Okay, what if we put a dead prey item at a displaced odor? Then again, it's going to the wrong place. So it's smelling the prey. And then if both were empty, then it wouldn't hunt in either place. So it's showing that it was just, if the live prey was there generating its electric field, it would ignore the scent and go where the electric field was. Whereas if the prey was electrically shielded, then it would divert to its olfactory system. So the electrical system was primary. If the electrical detection wasn't there, then it would go to the olfactory system.
That's a nice test. I like that.
So that's in the case of the platypus, the shark, and the lungfish are all doing passive electroreception. Right? They're detecting electrical fields created by their prey. For active electroreception, we do have several animals, particularly fish, that can create electric fields. And they do this with modified muscle tissue. And so these muscle tissue creates, so the same sort of thing that would create the electrochemical signal that would normally stimulate a muscle builds up like a stack of capacitors, those of you who do electronics, and build up an electric charge, and then they can release those electrical charges in bursts. So you build up electricity and then release it in bursts. And so these electric organs are located back here. And so since their swim muscles have been turned into electric muscles, they have to swim now by beating their fins back and forth because they can't swim with the muscles anymore. So here, for example, the elephant nose fish is freshwater African fish. And so its electric organ, again, this modified muscle, can generate electricity in these pulses. Right? It builds up electricity and releases them in these little pulses. Then, so that's how it creates electricity. Then it's got actually 3 different types of receptor cells that respond to electricity. The mormeromasts detect electric organs.
That's another one of those amazing words like glomerulus. Mormeromasts. Mormeromasts.
Discharge. So here they're creating an electric field and now are navigating by distortions in that electric field around them. So as the electricity flows in the water around them, if it goes through something more conductive, like a living organism, which is going to have ions in its body, it's going to be saltier than the environment around it, it's going to defocus those fields. If it's something inert, like a rock, it's going to spread them out. And so it can feel not only where things is, but is it biological or non-biological? The Nolan organ detects the electric organs of other knifefish. So not only its own electric field, but somebody else. So now this one it can use for communication. So this is what it's listening to the electric fields of other knifefish for. Then the ampulla receptors are like the ampullae on the sharks. These are detecting prey. Okay, so 3 different types of electrical receptors that are responding to different types of electric fields for different purposes. And so here's looking at, again, generating an electric field around itself and how that's destroyed. So again, a live organism tends to focus it because it's got higher ion content than the fresh water around it, where an inert object like a rock is spreading that out. So it can, by detecting distortions in the electric field, the proximity and quality of objects around it. And so here they're looking at that if you have a metal rod, depending on how close it is, so the closer it is, it creates actually a metal cube. It creates a stronger signal the closer it is. Whereas if you use a plastic cube, you're hyperpolarizing instead of depolarizing. And this, again, the closer it is, the stronger that signal is. So it's detecting both distance and quality of the object. And then the other way it detects distance is the closer it is, the smaller the receptive field, the larger it is, the bigger the receptive field that's being stimulated by the electric field of those distances. So detecting the position, distance, and quality of objects around it in these murky freshwater streams. And then in terms of communication, this is the knifefish. This is a South American fish. Again, uses undulations of its fin to swim because its muscles have been turned into electric organs. But when they're mating, they create these patterns of, we even call them like chirps, and as if a bird would be singing to each other. They create these certain patterns of communication that they use to find mates and to court their mates with. Okay, so using electrical reception both passively and actively. Now again, a sidebar to that, there are some animals that have used this ability to create electric fields not as a sensory system, but as a predatory system. And so electric eels and electric rays can create these really big jolts of electricity. We're talking 10 to 1,000 volts to knock out their prey. So their electric organs has like 6,000 of these electrocytes, right, electricity producing cells, these little modified muscle cells lined up like capacitors, and produce these 720 volts in the case of this electric eel, which is a freshwater fish. The electric rays are saltwater fish. In fact, we have a species of them off our coast here that are just ornery. They will sometimes just come up behind you when you're scuba diving and zap you for no reason. In fact, we had a group that was studying neurotransmitters at UC San Francisco, and and had us actually dissecting electric waves out of torpedo rays because they just were loaded with electroreceptors. You always knew if you had a hole in your glove because the organs would still fire when they were dissected out of the fish.
He always drops the craziest stuff, man. That's awesome.
Okay, so in addition to electric fields, there are also magnetic fields. And again, this is one of those things where somehow organisms from bacteria to whales are known to be able to respond to magnetic fields and to navigate that way. It's like having a compass, right? The Earth creates a magnetic field. If you tune into that magnetic field, you have a compass built in. And even bacteria and protists have this ability. How they do it, we have no idea. Now, typically we often find these iron oxide, these magnetite compounds in these animals, and we're assuming because it's magnetite, it's a magnetic material, it's like compass material. But there's been some studies saying, maybe not. So it's really, it's obviously widespread phenomenon. But again, we don't do it, so we haven't studied it so much. So an interesting way. So it is just absolutely amazing the varieties of sensory systems organisms have. That again, the complexity of how do you build a transducer, a cell that takes a physical signal from the environment and creates a generator potential. As complex as that is, there's these tremendous ways that organisms have figured out to do this. And that's absolutely amazing that there are so many ways that animals have found to respond to changes in their environment. So in our next lecture, we'll now look at the other end of this. So we talked about our responding to stimuli, basic things that organisms do. We've talked about the sensory input. We're now going to look at the motor output and look at how effectors, particularly how muscles, work in response to neural integration. And so we'll pick up our account from there.
Oh, is it called accommodation? No, it's not that. I think it's What the hell? Oh, you know what? It probably is gravity. I would guess it's low frequency. During an auditory transduction, across what membranes does ion flow vary? God, these images are totally crap. They've been compressed to all hell. And look at all these artifacts. This is JPEG'd out, bro. Come on. Hmm. This is not right. Uh, taco flavored tortilla chips. What the heck? Hmm. I hate when it just says that is incorrect. Because like sometimes it's like for dumb ones where it's like Na instead of salt, and it's like doesn't even make sense. There's no hint either. Okay, so these are right. As a result, the membrane depolarizes. The signal is perceived and disappears once the higher salt concentration. I think that the only other choice would be this. Bro, God. I don't like these ones. I'm gonna guess that this is still this. I hate these ones, dude. These ones are so dumb. Oh shit, they reversed it. The presence of glutamate triggers a GPCR-based signaling pathway and possibly secondary messengers. Because activated proteins and secondary messengers persist, the taste lingers long after even the presence of salt in the tongue causes sodium to enter the cells. As a result, the membrane depolarizes. The signal is perceived and disappears once the higher salt concentration is washed away. I'm going to try this one. That is so stupid. I hate those. Especially with no hints, those are the worst ones. I don't think they have 4 sets of eyes. Do they have... They might have compound eyes, but I don't know if that's... I'd guess it's probably excellent hearing. Okay, let's guess excellent hearing.
Let's review the structure and function of the parts of the vertebrate eye. The pupil is the opening that lets light into the interior of the eye. The iris is the structure that contains muscles that make the pupil larger or smaller. The muscle is the structure that contracts or relaxes to alter the shape of the lens. The ligament is a structure that puts tension on the lens, flattening it. The lens is the structure that changes shape to focus light on the retina. The retina is the location of the eye's photoreceptors. The retina is also the place where you would find rods and cones. The fovea is the place where photoreceptors are highly concentrated. The optic nerve is the structure that conducts action potentials to the brain. The blind spot is the place where the optic nerve leaves the eye. The sclera is the tough layer that forms the white of the eye. The cornea is the transparent part of the sclera that lets light into the eye. The choroid is the middle pigmented layer of the eye. The vitreous humor is a jelly-like material that helps the eye hold its shape. The aqueous humor is the liquid that helps supply nutrients to eye structures. Accumulation of this liquid can cause glaucoma. The lens focuses light on the retina by bending light rays, and it changes shape to adjust focus when you look at near or distant objects. When you look at a nearby object, muscles attached to the choroid contract, allowing ligaments attached to the lens to slacken. This reduces tension on the elastic lens, which becomes thicker and rounder, bending light rays more sharply into focus. This is called accommodation. When you look at a more distant object, the muscles attached to the choroid relax. This allows the ligaments to pull on the edges of the lens, flattening it. The flatter lens bends light less sharply, focusing light from the more distant object onto the retina. Let's review the structure and function of the parts of the vertebrate eye. The pupil is the opening that lets light into the interior of the eye. The iris is the structure that contains muscles that make the pupil larger or smaller. The muscle is the structure that contracts or relaxes to alter the shape of the lens. The ligament is a structure that puts tension on the lens, flattening it. The lens is the structure that changes shape to focus light on the retina. The retina is the location of the eye's photoreceptors. The retina is also the place where you would find rods and cones. The fovea is the place where photoreceptors are highly concentrated. The optic nerve is the structure that conducts action potentials to the brain. The blind spot is the place where the optic nerve leaves the eye. The sclera is the tough layer that forms the white of the eye. The cornea is the transparent part of the sclera that lets light into the eye. The choroid is the middle pigmented layer of the eye. The vitreous humor is a jelly-like material that helps the eye hold its shape. The aqueous humor is the liquid that helps supply nutrients to eye structures. Accumulation of this liquid can cause glaucoma. The lens focuses light on the retina by bending light rays. Let's review the structure and function of the parts of the vertebrate eye. The pupil is the opening that lets light into the interior of the eye. The iris is the structure that contains muscles that make the pupil larger or smaller. The muscle is the structure that contracts or relaxes to alter the shape of the lens. The ligament is a structure that puts tension on the lens, flattening it. The lens is the structure that
The vertebrate eye is similar in structure and function to a camera. At the back of the eye is a thin, light-sensitive sheet called the retina. The eye also has a lens that focuses the light onto the retina. As the eye looks at a point of light, the light passes through the transparent cornea and then through the lens, both of which bend the light, focusing it onto the retina. The retina is made up of a variety of cell types, including the light-sensitive cells called photoreceptors. There are two types of photoreceptor cells in the retina: rods, which are sensitive to dim light, and cones, which are sensitive to color. Each cone cell is most sensitive to one of three primary colors: blue, red, or green. The human retina contains mostly rods, but a region in the center of the retina called the fovea contains a dense concentration of cones. The fovea is the area of highest visual acuity. When people focus on details, such as a point of light, the eye moves to focus the image on the fovea. As light enters the eye, it must pass through several layers of neurons to reach the photosensitive area of the retina. When light activates a photoreceptor cell, the cells that synapse with the photoreceptor are activated, carrying a signal along the optic nerve to visual centers in the brain. Note that the rods and cones lie at the back of the retina, with their light-sensitive regions pointing away from the source of light. Rods and cones have segments that are packed with membrane-rich discs. The membranes of these discs contain large quantities of a transmembrane protein called opsin. Each opsin molecule is associated with a much smaller molecule called retinal. In rod cells, the two-molecule complex is called rhodopsin. Each of the three types of cone, blue, red, or green, has its own specific kind of opsin. The retinal molecule is the light-sensitive portion of the complex. When retinal absorbs light, it changes shape. The change in the shape of retinal causes the surrounding opsin to change shape. The opsin, in turn, triggers a cascade of events that activates the cell. The rods and the three types of cones each have different opsins that determine the wavelengths of light that the retinal will best absorb in these cells. Rods are more light-sensitive than cones and are able to work under low-light conditions. Cones respond mainly to bright light and are responsible for color vision. For example, the type of opsin retinal complex in the blue cones responds best to blue light. It also responds to yellow light but less strongly. Intermediate wavelengths cause graded responses. Information from all of the photoreceptors enables the brain to differentiate many hues, using only three main types of color photoreceptors. The brain combines the signals from the three cones in much the same way as the three primary colors of light can be mixed to produce all the colors of the visible spectrum.
Response of a photoreceptor cell to light. Human vision begins with the detection of light by photoreceptors, rods and cones, which are sensory cells located in the retina of the eye. The detection of light triggers sensory transduction, a chain of events that conveys information from the sensory cells to the brain. Here we'll focus on rods and explain how they transduce light energy into a change in membrane potential called a receptor potential. A rod is an elongated sensory cell that has its cell body and synaptic terminals at one end, and a compartment known as the outer segment at the other end. Sensory transduction occurs in membranous discs that are stacked from one end to the other in the outer segment. Let's zoom in on the outer segment to examine the molecular events in sensory transduction. First, we'll look at a rod cell in the dark. This figure shows a cross-section of a small part of the outer segment. Some of the key molecules shown here are located in the disc membrane, while others are located in the plasma membrane. In the dark, the proteins rhodopsin, transducin, and phosphodiesterase are inactive, and the cytosol contains a high concentration of cyclic GMP, written as cGMP. Molecules of this cyclic nucleotide bind to the sodium channels embedded in the plasma membrane, keeping these channels open. As a result, sodium ions diffuse freely across the plasma membrane with a net flow into the cell. As the graph shows, the continual influx of sodium ions results in a steady membrane potential of about -40 millivolts. This is a much smaller membrane potential than the -70 millivolts typical of many resting neurons. In this depolarized state, a rod in the dark continually releases the neurotransmitter glutamate. What changes when we step into the light? Light reaching the retina strikes rhodopsin, a visual pigment embedded in the disc membrane. Light converts a component of rhodopsin, cis-retinal, to its isomer, trans-retinal, which has a different shape. This shape change converts inactive rhodopsin to active rhodopsin, kicking off signal transduction. Active rhodopsin activates transducin, a G protein. Activated transducin dissociates from rhodopsin, diffuses along the disc membrane, and binds to phosphodiesterase, an enzyme. This binding event converts phosphodiesterase to an enzymatically active form. Active phosphodiesterase, in turn, begins hydrolyzing molecules of cyclic GMP in the cytosol, converting them to GMP. As the level of cyclic GMP falls, fewer and fewer sodium channels have bound cyclic GMP. Without bound cyclic GMP, sodium channels close. How does the closure of sodium channels affect the permeability of the rod's plasma membrane? The closing of sodium channels in the plasma membrane decreases the membrane's permeability to sodium, preventing further diffusion of sodium ions through that channel. Now let's look at the graph of membrane potential for this scenario. In the light, as sodium ions stop entering the cell, the membrane potential becomes substantially more negative, as this graph shows. Hyperpolarized, the rod cell stops releasing the neurotransmitter glutamate. Sensory transduction in rods involves elements found in many signal transduction pathways, including receptors, G proteins, membrane-associated enzymes, second messengers, in this case cyclic GMP, and ion channels. Activation of the transduction pathway in rods leads to a change in membrane potential, which affects communication between rods and other cells in the retina. In the rod cell, this change, called the receptor potential, is a hyperpolarization. Note that this differs from the receptor potentials of most other sensory receptors, which are depolarizations.
Remain closed? No, that's not right. All right. Good stuff. Homework's done.